Good evening. My name is Chuck Natanson. I'm an intensivist, not a statistician. And I think the reason that I give this lecture is I've written quite a few meta-analyses, some of them quite controversial. And what I'm going to do today is first present some background information of how you approach and write a meta-analysis. But that's not going to be the major focus. I'm going to describe some of the meta-analyses that I've done with the idea of trying to bring to life the terms of I-squared, meta-regression, fixed random effects models. So the terms have real meaning. And in the end, when you read a paper and it's a meta-analysis, you'll understand those terms. Hopefully, I can get that across. But let me begin with the first half of the talk, which is a discussion of what are the major parts of a meta-analysis and how you do it. So the term meta-analysis was first coined in 1976. And you can say, well, why in 1976 did they first try to define a meta-analysis? Well, in some respects, a meta-analysis is dependent upon a randomized controlled trial. And the randomized controlled trial was really invented in the 1930s by a man named Fisher. And so it took about that many years to have enough randomized controlled trials in certain areas in order to need to combine them. And the term as defined is the statistical analysis of a large collection of results from individual literature for the purpose of integrating their respective findings. There's really two types of meta-analyses, and the second one is the one I do more of. The first one, and I think that's done less today, is to determine if there's a similar treatment effect exists for a therapy in independent studies to estimate a net effect for this therapy, with the idea that you did a large number of randomized controlled trials and they were underpowered. And when you combine them, you'll find a significant effect. And one can say is can you use this to approve a drug by the Food and Drug Administration? And my statistician that I worked with for many years sort of said this. He believed that a properly done, that's the correct terms, and we'll go over that, meta-analysis may well be equivalent to a multi-center randomized controlled trial. But in the end, he said he would believe more a multi-center randomized controlled trial. The second purpose of a meta-analysis, which is what I use it for, is if you find treatment effects differ substantially for a therapy among independent studies, is try to examine factors that may explain these differing effects. Some trials may have showed something was beneficial, and other trials showed they were harmful. And if you do something we'll go into called the meta-regression over certain factors, and we'll give you examples of this, you can determine what was the cause. And this can give you great insights into what was the history of this therapy and why in some instances it was beneficial and why it was harmful. And I find this type of meta-analysis much more interesting. So I'm going to go through the 4 or 5 steps of doing a meta-analysis. The first one is formulating the question. You have to have an interesting question or one that makes sense because the validity and importance of the study are contingent upon this step. And a poorly conceived research hypothesis will usually lead to an analysis of dubious value. The second thing is you have to define prospectively. A meta-analysis should be done prospectively. The only thing different from a randomized controlled trial is the patients have already been enrolled. And you have to write out ahead of time your inclusion criteria. It should be prospective, systematic, and explicit. And ideally, you want to combine randomized controlled trials with a similar diagnosis, outcome, patient characteristics, and treatment groups. So there's two issues here with how you combine data and whether you should only combine randomized controlled trials or you should try to analyze observational studies. The advantage of including all the available studies, regardless of the size, design, or quality of results in analysis, is that it's broadly representative of the clinical experience. But because of this, it may compromise the accuracy. Alternatively, exclusion of poorly done studies may increase the statistical validity, but limit the ability to generalize the results. If you only use very consistent, randomized controlled trials, you may not get the full spectrum of what has happened. What I actually like to do is include them both, observational studies and randomized controlled trials, and analyze them both and see if they have similar findings or different findings, so you get the full clinical experience. At the same time, you get the data which has the most statistical validity. So, a meta-analysis, after you have your question, it usually begins with a search, and most of the time you have to go to a professional that are available in libraries, and you search databases such as Medline, Current Contents, Best Evidence, Cochrane, and HealthStar, and you have search terms, and then you get a large number of papers usually, and you search the title abstracts to figure out which one of the studies are pertinent to what the question you were asking. And then you go through those studies which are pertinent, which they fit the design or the question that you're asking, and have the appropriate data. You go through the full texts of those articles and you review them thoroughly. And you go through the reference lists to see if there's additional articles. And once a study is selected for inclusion, the data should be extracted from that study by more than one reviewer into structured forms which have your prospective analytic plan in them of what things you're going to analyze. The fourth step is you've got to find a common measure that you're going to use for your treatment effect. And I'll go over that. You have to, because you want to include as many studies as you can. So you want to define your outcome as loosely as you possibly can, or as specific if you want to narrow it down. The next issue is, should you do a fixed versus random effects model to combine the data? And this is important when you look at papers. There are just two ways of analyzing the data and combining them that have different sources of variability. A fixed model, in general, really is to try to tell you or make conclusions about the studies. A random effects model is a larger source of variability. It's try to make conclusions about the patients. And in general, I think you should do both a fixed and a random effects model and present both. And most of the time, they will not disagree. But there are certain situations where a fixed will be more accurate than a random effects model. Now, the other important thing to know, not only whether they use a fixed or a random effects model to analyze or combine the data, is the Q statistic or I-squared calculation. Now, I don't know actually how to calculate an I-squared, but I know how to interpret it. The I-squared goes from 0 to 100%. And when you read a study and you look at the I-squared, if it's below 20 or 30%, you feel comfortable combining the data. So what does the I-squared mean of 0 to 20%? You would ideally like it to be 0%. And when it's 0%, the fixed and random effects model are identical. Now, what is it telling you? It tells you that the variability within the studies can explain the variability across the studies. So when your I-squared is small, it means the variability within a study explains the variability across the studies. When the I-squared is 30% or greater, that means that the variability within the studies can't explain the variability across the studies. And what am I saying by that? Well, I think you can see where one says when you have an I-squared of greater than 30%, I'll give you this example. So you're going to have a high I-squared if some studies show significant harm and some studies show significant benefit. Would it make any sense to combine those together and say there's no effect? It makes a lot more sense to do something called a meta-regression. Try to figure out what factor, and we'll go over this later on, in those studies explains why some studies are beneficial and some studies are harmful. And you really, when you have apples and oranges, you really shouldn't combine studies. Now, that isn't completely true. What I was talking about is a qualitative interaction. That's the term that's used. That means some studies are on one side being beneficial and on the other side being harmful. You should never combine those kind of studies. And you shouldn't believe a meta-analysis which has a high I-squared. But if some studies are only slightly harmful and other studies are really harmful, it's probably okay to combine them and try to interpret why the studies, in general, you're making a conclusion that the studies are harmful. It's just you can't determine what the level of harm is, which usually is not that important, or the level of benefit. The next issue is when you do a meta-analysis, you've got to examine for bias. When you have a beneficial or a harmful effect, there should be a normal distribution of the study's effect sizes around that effect. And I'll go over what meta-regression is with examples, a funnel plot and publication bias, what the I-squared is, and hopefully those terms will become very easy then to understand as opposed to the dry way of presenting it now. The fifth step in reporting, interpreting results is they've gotten together and made sort of rules that they've published in papers. And to improve the quality of reporting for meta-analysis, they have checklists and flowcharts they've published. It's called the quorum statement. The quality of reporting of meta-analysis, they provide guidelines for reporting searches, study selection, validity assessment, data abstraction, study characteristics, and data synthesis. And when you try to publish a meta-analysis, they ask you to give those checklists and show how you did it and to try to improve the overall quality, that you followed the prospective methodology. So now I'm going to change the focus. Now let's do a number of meta-analysis that I've published previously in papers, and we'll go over what an I-squared, what meta-regression is. And I'm a sepsis researcher, and I take care of patients with septic shock. And the first thing I'm going to go through is the meta-analysis that I published on clinical trials in sepsis. Septic shock is a bacterial infection, and the management is usually early recognition. You try to have a high suspicion. You want to give the right antibiotics right away. You want to do, these people develop hypotension. You want to give fluids. Their blood pressure is low. You want to give agents which will cause if their hypotension is severe enough, you want to give agents which will cause the blood vessels to constrict so the blood pressure is maintained. And if there's a site of infection, you want to remove it. And to show you, sometimes you don't need an analysis. This is an analysis. What I'm showing you here is a Christmas tree. And I'll be showing you this throughout the talk. This is a classic Christmas tree. This is the no effect line. These are the odds ratios of survival, which is probability of surviving in the treatment group over the control. And here would be early versus late antibiotics. These are 95% confidence intervals. If the odds ratio lands here and it doesn't touch the no effect line, there's a significant beneficial effect. These are all how meta-analyses are presented. And this means your odds of surviving are 10 times greater if you've got appropriate antibiotics, which means that the cultures showed that you were on the appropriate antibiotics, versus inappropriate antibiotics, that the cultures showed that the antibiotics you started were not effective. And you can see here you don't need a meta-analysis, that no matter what year the study was done and the author, that all the studies showed the same effect, and that there's a benefit of giving the appropriate antibiotics. And this is the benefit of giving antibiotics early versus late. And once again, you can see the odds ratios of survival here. You don't need an analysis. No matter what the diagnosis was, the year of the study, you found a beneficial effect when you gave early and appropriate antibiotics. So now let me show you a meta-analysis where you did need to do an analysis to figure it out, and you had to do a meta-regression analysis. So in the 1980s, people had the idea that septic shock was caused by the bacteria stimulating inflammation, that you had an infection, the bacteria released pathogens and toxins, endotoxin. Your host system was stimulated to release inflammatory mediators, and it's the host's own inflammatory mediators which caused the shock and injury. And it led to multi-organ failure and death. Now, in the 1980s, there were huge numbers of clinical trials, and the biotech industry just was emerging at this time, and multiple biotech companies folded because they did these clinical trials, and it turns out that they couldn't confirm this hypothesis, that they used anti-inflammatory agents in patients with septic shock, and they couldn't find a beneficial effect, and sepsis was considered the Bermuda Triangle of the biotech industry. So the first question that we asked is, if you looked at all of these anti-inflammatory agents together and you did a meta-analysis, could that help us figure out why these clinical trials failed? So this is my first meta-regression. What I'm regressing this meta-analysis against is the size of the clinical trials from 0 to 2,000 patients. And again, here's our no effect line. This is the odds ratio of survival, which is the probability of surviving if you got an anti-inflammatory agent in your septic versus a control population that was treated by all the normal treatments except they didn't get an anti-inflammatory agent. This is your 95% confidence interval. This means that if you got the anti-inflammatory agent, you were 2 times more likely to survive, 4 times more likely to survive, 8 times more likely to survive, and here you're more likely to die. So these were trials of anti-tumor necrosis factor antibodies. Tumor necrosis factor is released by white cells. It's a pro-inflammatory cytokine which causes inflammation. And what you can see here is these are all the trials, and this trial was 2,000 patients, and there was no benefit. The odds ratio crosses. There was no benefit here. The 95% confidence interval crosses the no effect line. You can see here in a trial of 1,000, you find the same thing. Here you find, again, the same thing. And again, in this trial of about 500, you find the same thing. But when you get below 500 patients, these small trials, you really find now it's on both sides of the no effect line, and there's no effect. But there seems to be a small trend that's very similar when you have trials over 500 patients, at least for this anti-TNF antibody. So let's look at the next treatment, soluble TNF receptor. Large trial again, a small beneficial trend. And shown in white here, I now have turned the antibodies are blue, and now the new treatment, the soluble TNF receptors, are white. And you see the same beneficial trend, and when you get to smaller trials, the effect is more variable. And if I look at IL-1RA, which is another anti-inflammatory agent, I found the same trend. And PAF receptor antagonist, another anti-inflammatory agent, the same trend. And antiprostaglandin, again, we found the same trend. And antibradykinin, again, the same trend. So you don't really need an analysis because you see the large trials, the top 12 trials, all showed the same beneficial trend. And so what it suggested is that the mistake we made is that this isn't such a beneficial treatment. It's a very minimally beneficial treatment. It only has a 1 or 2% increase in survival. And when you get to trials below 500, you see this variability because no longer do you have a large enough sample size to capture the trend. And it wasn't that they failed, they overestimated the treatment effect, and this therapy really, most people come to believe, is not very effective. It's minimally effective. But the question we asked, the next one, is why did all these anti-inflammatory agents, what was common about them that they would all have the same effect? And we looked at a lot of different things. And the one thing that we found, which was very similar over all these clinical trials, was the control mortality rate. The IL-1RA, the antibiotic and the PAF, and this is very unusual, they all had about a mortality rate in the 30s. So the control mortality rate was very beneficial. Was very low, about 30%. And so the question we asked, so you have the small effect of about a 2% benefit done at the same control mortality rate at 30%. So all of these trials studied animal models. And in the animal models, all these anti-inflammatory agents were very beneficial. There was 95 different animal studies that were done. So we asked, what was the control mortality rate that they did in the animal models? Was it the same as the control mortality rate that was done in these human clinical trials? And so in the next slide, I'm going to show you data from the 95 animal studies that were done, that were referenced in these clinical trials, and I'm going to show you the control mortality rate, and I'm going to show you the treatment effect, and I'm going to do, again, a meta-regression. So now here again, I've just turned the Christmas tree on its side. This is the no effect line. This is benefit, 10 times more benefit, increase in the odds ratio of survival, 100, 1,000. And here is more harmful. And each one of these, the size of these circles is proportional to the number of animals in these clinical trials. And the first thing you notice when you look at this is almost nobody did a clinical trial below a mortality rate of 50%. This is the odds ratio of dying. I'm using this to not constrain the range. But 1 is a 50% mortality in the control group. This is 100% mortality in the control group. This is less than a 1%. And you see at a 50% mortality, there's almost no studies. There's only these two. And they had very low control mortality rates, and they were harmful. Well, when I went to companies and I gave this talk, the scientists there would all say to me, oh yeah, we knew that. Anytime we used a low mortality rate model of infection, and not a severe infection, the agents were harmful, and we threw the data out. It's the first thing. Next thing is, what you see is, despite all these studies being above a 50% mortality rate, there's still a relationship, and it's highly significant. And this is beneficial, this is harmful, but as you go, even with this constrained range, from a high mortality rate in the control group, there still is a significant relationship, and as you go down, the treatment effect gets smaller. So now, let's go back. We've done this meta-regression, and it sort of suggests control mortality rate is important. Let's plot the human data on this graph. Where would it fit? It was done at about a 30 to 40% mortality. Well, it fits pretty good, exactly where you'd expect it. The treatment effect at this much lower control mortality was much lower, as the animal data would suggest it. So the next question you would ask is, all this is retrospective data. Can I do an experiment prospectively and test this hypothesis that the control mortality, that the treatment effects in the human clinical trials were very small, but the control mortality rate was very similar at 30%, and it's only at high control mortality rates of 70, 80% that anti-inflammatory agents are beneficial. So what we did was we took animal models and we gave them different doses of different types of bacteria and toxins to produce different mortality rates. to test if our hypothesis prospectively, at least in animal models, was true. And as you see here, we had different size studies. Again, this is the no effect line, benefit, harm, the control odds of dying. And what you see here is if you're above a 50% mortality, things look pretty good. But if you're below a 50% mortality, which would be here, things don't look so good. And again, there's a significant relationship with these anti-inflammatory agents in these models and using different types of treatments and different types of bacterial challenges. And if we did it in a large animal model, we found the same thing. And if you plot the human clinical trials, it fits exactly where you'd expect. So in summary, using meta-regression and meta-analysis techniques, which I hope is much clearer now, we were able to figure out, we think, that the treatment effects were very small in these clinical trials done in humans, but statistically significant. Using meta-regression, we determined that the beneficial effect of anti-inflammatory agents is dependent upon the risk of death or the control mortality. Beneficial at high risk of death because when you have a very, very severe infection with a very severe inflammatory response, that inhibiting inflammation overall has a beneficial effect. But if you have, it's ineffective or harmful when risk is moderate or low. When the inflammatory response is working well and you're going to survive, it's not beneficial to inhibit it. In fact, it'll increase mortality because you're blocking the inflammatory response. So this is a very difficult treatment to give and a very difficult treatment to use clinically. So I'm going to do another meta-regression. Corticosteroids, those are steroids that have been tested in septic shock for over 50 years. And I'm now going to do a meta-regression to try to figure out what was the effect of these steroids in sepsis over the last 50 years. They've been investigated since the 1960s. In the early 1990s, they were shown to be ineffective or harmful, but there became a renewed interest after the 1990s in lower doses in septic shock. So now, again, here's my no effect line. This is benefit, this is harm. I'm going to be looking at the odds ratio of survival, and now I'm going to do a meta-regression across the year the study was published, because that's the factor which is informative. Before I looked at control mortality, now I'm going to look at the year published. So these were the studies that were published from 1963 to 1998, and they used very high doses in short courses, and kind of focus on this one study. So remember I told you want to look at the I-squared when you do a meta-analysis. And here it's 70%. So you would say, I'm not going to combine this data because this study is showing benefit and this study is showing harm. And you say, okay, what is it about this study? Because you can't just exclude a study based on a statistical analysis. You have to have a methodologic reason. Well, this study was a single center, unblinded study done by a single surgeon over 10 years, and he was running an observational study and a randomized controlled trial at the same time. I'm sorry to say that we actually changed our care and started doing this treatment based on this study at the NIH. We started using these high doses of steroids. But if you exclude this study, which was done very differently, these were all mostly multi-center randomized controlled trials, the I-squared goes to 0. The variability goes away, and you find actually that high-dose steroids, short courses, actually is harmful. So I hope you got the idea of what the I-squared tells you. Again, meta-regression by years. So short courses of very high doses of steroids, this is a huge dose, equivalent of 24,000 milligrams of hydrocortisone, worsen survival. So 10 years later, mostly in Europe, people started studying them again. And you find now that there's a very different appearance than this one. And there's a significantly beneficial effect now. And the first thing you should ask me is what the I-squared is. So stress dose steroids, much lower doses, tapered over 6 days, given over longer courses, improve survival. But what's the I-squared? It's 25%, so it's borderline. But you still are worried about it because you'd like the I-squared to be 0, but it may be acceptable. But it turns out that it's probably being caused by this trial. Because if you remove it, the I-squared goes to 0. But whether you remove it or not, there's still benefit. So you need to do some investigating here. And so the first thing we did was, since steroids are sort of anti-inflammatory, we went back to our control mortality as the meta-regression. So why is the Corticus study different from the 11 other trials? And remember, it could be the Corticus is right and the other 10 trials are wrong, or they both could be right, but they're showing different effects, so you sort of want to try to figure this out. So when we graphed this against the control odds of death, we found this same relationship again, but which one's the Corticus? Well, it was studied at a very low control mortality rate, and turns out it's not unexpected what they found. Well, this is a large, multi-center, randomized controlled trial. And these are relatively small trials. They're 40 or 50 patients. And this one, multi-center, randomized controlled trial, you sort of believe, and it sort of suggests to you that at least at low control mortality rates, there's no benefit of steroids in not such sick patients. But in very sick patients, this data suggests, well, maybe it's beneficial. Well, these are small studies. So now we need to look at the issue of publication bias, because if you have a small study, people tend to only publish them if they're beneficial. If they're not beneficial, they call it the drawer effect. They tend to not publish them. So what we need to do now is a funnel plot. And that's what I discussed with you in the beginning. What is a funnel plot? Okay, let's go through that, because that will tell you if there's a publication bias. So here is the no effect line, and precision just means the size of the trial. And here's the log odds ratio of death. I'm sorry this is reversed. Now control is here and steroids are here. I should have reversed this, but I haven't done it yet. And let's now look at the treatment effect. So this says that steroids in this meta-analysis of these these lower doses was beneficial, and here's the treatment effect. And there is a small beneficial effect on survival. So basically, what you're going to look at is what's the distribution of the studies around this effect size, because they should be equally distributed on both sides of this effect size. And when you look at it here, what you find is there's these bunch of small studies that are way over here. Now, these are the studies that are 40 or 50 patients, and this is what's driving the beneficial effect. And you can actually plot, and this is why it's called a funnel plot, where you'd expect studies based on the treatment effect to reside, and that's what this red line shows. So the studies should be within, in order for this effect to not be biased, should be normally distributed, all these dots should be equally distributed in here, in this funnel. And what you find is 1, 2, 3, 4 of these small studies are prejudicing the data and saying it's benefit. And you would have expected there to be equal number of studies over here if this was truly an effect. And what you believe is these are the studies that are sitting in somebody's drawer. They weren't beneficial, they were small, they didn't think anybody would publish it. So that's what publication bias is, that's what a funnel plot is, and that's how you determine it, and that's why you do it. Because the small studies are on the bottom, large studies are on the top, okay? And you can see that there's a preponderance of small studies that are over here and not over here, and that's the publication bias. So do corticosteroids have any effects that are consistent across these clinical trials, different from survival? And it turns out reversal of shock, or reversing the bad blood pressure, occurs earlier if you give steroids. And this is a very consistent effect. What's the I squared? It's 0. And all the studies that reported it found the same thing, so this effect you'd believe. So let's summarize this and what we learned over 50 years of data. And what we learned is despite 50 years of research, we still don't know if steroids are beneficial or not. We only know that they reverse shock. So its effects during sepsis depend on dose and severity of illness. High doses are harmful, and they seem to be more beneficial in low doses in very sick people. High-dose corticosteroids increase mortality. Low-dose improve survival in severely ill patients. Low-dose steroids decrease vasopressor requirements and enhance or reverse shock. But at present, we can't really know what's going on because the beneficial effect of low-dose steroids are based on small trials with only 40 patients, 40 to 44, and they're confounded by publication bias. The largest trial we actually can believe, and it tells us in not such sick patients that corticosteroids are not beneficial. And at present, until a large multi-center trial tells us whether or not in very sick patients corticosteroids are beneficial, you really don't know to use them or not. You have no data except that it will reverse shock. So until new data are available, you have to use clinical judgment. And what is the risk-benefit to administer low-dose steroids for septic shock? Because it has to be individualized based on the severity of illness. If you think somebody's very sick and they're going to die, it will reverse shock. But then steroids have risks. They can increase the risk of infection. They can immunosuppress you, and you have to decide what that risk is versus the benefit, the potential reversal of shock, and there may be a survival effect. So what I'm trying to say to you is by analyzing the data, looking and doing meta-regression, meta-analysis, looking at publication bias, meta-regression, we're able to define this data and figure out what it shows. So let's look now at another set of studies, and this is sort of a sad thing in the medical industrial complex, what happened with intensive insulin therapy. So they had some people in Belgium, an anesthesiologist, had the idea that we should, like diabetics, if you keep them under very close control, there's a belief that they get less vascular damage. So let's take somebody with shock or severely ill, and let's keep them under very tight control, and let's see if that has a benefit, like the benefit you see over 20 to 30 years when you treat somebody with diabetes. And it was published in the New England Journal, and it showed a benefit from keeping the glucose below 100 in people with shock. And this clinical trial was endorsed, single, unblinded trial, like the steroid trial I told you. Single, unblinded trial caused the Joint Commission on Hospitals to adopt tight glucose control, the American College of Endocrinology, the Volunteer Hospital Associations, the Institute for Healthcare Improvement, the Surviving Sepsis Campaign, everyone based on this single center unblinded trial adopted this therapy. So let's look using our different methods, let's look at this study a little more carefully. Who did they study with this tight glucose control using high doses of insulin? Well, really the majority of the patients, 60%, 63% were cardiac surgery. So who they studied is mostly people with coronary disease having placing cabbages or placing veins in the coronaries to increase the flow. And these people usually go home in 1 or 2 or 3 days. This is an operation that has a very low mortality rate, 1 or 2%. And who did they find that they benefited in this study? Well, it was mostly the cardiac surgery. This is the number of deaths, the difference in deaths between intensive insulin and conventional. And you can see the difference in deaths was mostly in cardiac surgery and thoracic surgery that made up this beneficial effect that they found on survival of keeping the glucose very tightly controlled between 80 and 90 in patients with cardiac surgery. So it was a single-center, unblinded study. And the thing that's funny about it is it had a relatively high mortality rate. In the state of Massachusetts, there are 11 intensive care units. If any of them had this high a mortality rate, they would all be shut down. This is 5%, is more than double the normal mortality rate for this procedure. The other thing that was funny about this study is they wanted to give insulin. So what they did is they took the patients postoperatively, and as soon as they came, these are people that are going to go home in 2 or 3 days, and they gave them high glucose loads. These are enormous doses of glucose so they could give them insulin. So this really wasn't a control. This was really the control kind of treatment. And high doses of insulin and high doses of glucose can give you infections. And it's not routine care. And so I guess what I'm saying to you is if you had people with coronary heart disease and you gave them something to raise their blood pressure, which would increase their risk of a heart attack, and gave some of them something to decrease their blood pressure, and you found that that was beneficial, you really wouldn't call it beneficial. That's sort of what they did here. They took people and made their glucoses high, and they treated them with insulin, some of them. So once this study was published, and most of these things take about 10 years to reverse, you can see here now that there were multiple other studies done. And nobody could reproduce this effect. This is the study I showed you. Nobody could reproduce this effect. And in 27 trials of tight glucose control, the I-squared is 17%. There's 8,000 patients. And this is acceptable. And overall, there was no benefit. So now what I'm going to show you, and the purpose of this, is discuss sensitivity analysis. When you do a meta-analysis, you want to do a sensitivity analysis, and you don't really care if there's a significant p-value, but you want to see if the effect size is consistent, because you want to make sure it's not due to one particular type of critically ill patient. And I'll show you now what a sensitivity analysis is, which most meta-analyses were done. The purpose is to show the effect is consistent over the different types of trials you did. So if you look at patients that had glucoses of less than 110, that is, they didn't have so tight, or rather, these are the very tight control, the effect size was the same, no benefit. If you look at the less moderate control, 150, there's still no benefit. If you look at surgical ICUs, there's still no benefit. If you look at studies done in medical ICUs, there's still no benefit. If you look at med-surgical, there's still no benefit. So this was really not beneficial no matter where you looked. And really, they couldn't reproduce this original study's effect. So what was the problem, or why was there a harmful effect? And what you found is in those studies that reported it, whether the glucose was less than a 10, you got hypoglycemia. If you kept it less than 150, you still got hypoglycemia. And this is sugars less than 40. If you used surgical ICUs, it was less than 40. Medical ICUs, it was hypoglycemia. Med-surgical ICUs. So overall, the I-squared is 0. What happened in these studies is there was a risk of hypoglycemia, which increased or made the outcome worse. So in Australia, they decided to do a very large study and to compare tight glucose control in this study of 6,000 patients in medical ICUs. So there'd be a single study that was done in Australia, New Zealand, and Canada. And this study found that tight glucose control increased 7 to 8-fold the risk of hypoglycemia independent of the target's glucose or type of ICU, medical or surgical combined. So this paper, almost 10 years later, is published in the New England Journal. They do 6,000 patients, and they test this tight glucose control. And you can see they enrolled 6,000 patients, 3,000 in intensive, 3,000 in conventional. And these were surgical patients, 37%. This means that 31% were very sick, 21% had sepsis, and most of them were on mechanical ventilation. And what they found is tight glucose control significantly increased mortality. Just like with high-dose steroids. Same problem. In 6,000 critically ill patients, tight glucose control associated with hypoglycemia and increased mortality risk in 90 days. On the basis of this, we do not recommend use of lower targets, less than 110 in critically ill patients. So, I've been doing meta-analysis for a long time, and I've been doing these studies in the field of sepsis, and sort of what went wrong with these steroids and what went wrong with the tight glucose control. It's actually quite a pattern. If I look at all the trials that were done, the meta-analysis, there was one significant beneficial effect, and then the subsequent trials is what I'm going to show you in the next analysis, and to try to explain what went wrong. So, in sepsis, they tried anti-endotoxin. Endotoxin is a part of the bacterial cell wall. And one study shown here, published in the New England Journal, they took firemen and they injected them with endotoxin and took their serum, which would have antibodies to endotoxin. And they published a paper in the New England Journal that said that this was significantly beneficial. Well, so this is, as I've shown you, benefit, harm, the no effect line. And you can see all the subsequent people that tried to reproduce this result couldn't reproduce it. Well, they also tried a monoclonal antibody against endotoxin. Again, published in the New England Journal, significant beneficial effect. And you can see subsequent trials and an earlier trial couldn't reproduce this effect. And when you combine all the blue ones, there's a p-value of 0.6 that couldn't reduce the red one that was significantly beneficial. And here, 0.59. High-dose corticosteroids, we went through that. This was that study by the surgeon. And all the subsequent studies couldn't reproduce it. In fact, subsequent studies showed harm. Here's IL-1RA, a beneficial effect, couldn't be reproduced. Activated protein C, this is an anticoagulant that they used in septic patients. It was published again in the New England Journal. Subsequent studies couldn't reproduce this beneficial effect, p-value 0.5. Intensive insulin, again, published in the New England Journal. Again, beneficial effect, and I just showed you this data. Subsequent couldn't reproduce it. And again, if you put these all together, it's 0.32. And if you look at the shift from the beneficial to the last study, there's a significant shift. And so what do I think went wrong? Well, most people think that when you randomize, magic has occurred. And that suddenly you have data that is very interpretable and very important. But all randomization does is get rid of one form of bias, selection bias. And what really is important in science is reproducibility. The randomized control trial minimizes bias but does not eliminate the need for reproducibility. And I think that's what happened in these clinical trials. It's the sine qua non, the indispensable and essential condition of science and of scientific evidence if your findings are reproducible in other people's hands. So I hope that with my example of sepsis in clinical trials and a little bit of I gave you before, you now know what a meta-regression is, what publication bias on a funnel plot is, why you do sensitivity analysis, what the I-squared means, and how you interpret an I-squared and how it helps you analyze a study. I now will open this up for questions, if there are any, about what I've just presented. Thank you. Shoot. You need to speak loud. I'm old. And
Going back to the sepsis and steroid study,
you want to use the microphone, or you don't have to if you want to just stay seated? But then I gotta move forward here.
Sepsis and steroids. The timeline is kind of interesting. You go over extended period of time, and earlier trials show different effects than later trials. How do you control for changing manufacturing formulation of the drug? Is it the same drug they used in early studies and late studies?
No, that's one of the, no, well, most of them used hydrocortisone, but that is a weakness in the study. But we didn't find that when we looked within the different groups, we didn't find there was variability, that the I-squared was acceptable, so it didn't seem to be a cause of variability, so we thought it's okay to average over those studies. That's why you do an I-squared. At least statistically, we couldn't find a difference in those treatment effects. But you may want to do sensitivity analysis, which we now know what it is, and look at the different manufacturers and see that the effect is the same using a sensitivity analysis, so that it isn't just one type of manufacturer of steroids that's causing the problem.
Yeah, because we all know that quality control and manufacturing changed.
Sure. And you may want to do that with sensitivity analysis to see if any one of the manufacturers made a difference. That's why you do sensitivity analysis. When you do the I-squared, it says it's okay to put it together. And when you looked at the studies by the year, the meta-regression, I'm trying to bring this back, that helped you understand what the early studies showed and the later studies shows when you regressed it against the year the study was done. Yes?
I thought the use of control mortality as a dependent variable for the meta-regression was interesting. So I was wondering, in determining what you regress for, does that have to be determined beforehand, like prospectively, or do you decide kind of as you go what things you think are impacting the odds ratios that you're seeing in different studies?
So the way I'm answering this is the way my statistician taught me, and what he did was based on his interpretation of R.A. Fisher. So this is a different issue in statistics, but when you have a significant effect, you have a right to go searching for the cause. So as long as you find that the heterogeneity is significant, that the I-squared is very high, you do a test for variability and it's significant, to me that gives you a right to then go fishing what the cause is. If you don't find a significant p-value and significant variability, you have no right to go looking, and then you have to start correcting p-values. And so once you find heterogeneity and it's significant, you're, I believe, duty-bound, and you look at different factors to see if something explains it. Because you're trying to explain a significant effect, and I think in that situation, it's very different than when you have no significant effect and you're trying to explain results. Okay then, thank you very much for listening.